Welcome back. So we have just introduced our different cytoskeleton proteins. We've talked about microtubules, microfilaments, and intermediate filaments. And now we're going to go into them a little bit more in detail. So first we're going to study the microtubules. They're the largest structural elements in the cytoskeleton. If you remember, they are wrapped around here with a pretty large diameter, 25 nanometers across. They're involved in a lot of different functions in the cell. Mainly involved in movement, moving components within the cell, moving fluid outside of the cell. There are two different types of microtubules. The first one is the cytoplasmic microtubules. Cytoplasmic microtubules go all through the cytoplasm, and they do things like maintaining axons, forming mitotic and meiotic spindles, maintaining the cell shape or changing the cell shape, helping vesicles be properly placed and moved around, and orienting cellulose microfibrils during plant cell wall growth. The second type of microtubules are the axonemal microtubules, and these include organized and stable microtubules. They're found in structures like cilia, flagella, and basal bodies that the cilia and flagella attach to. Tubulin are made of heterodimers, and these heterodimers and alpha and beta, these two components, are the building blocks of microtubules. Microtubules are straight and hollow cylinders, and they are made of protofilaments. And the subunit of a protofilament is a heterodimer, one alpha tubulin and one beta tubulin. And these two alpha and beta bind non-covalently to form an alpha-beta dimer. And this alpha-beta dimer is pretty permanent. Normally, it does not dissociate from each other. Microtubules form by reversible polymerization of tubulin dimers. So all of these tubulin dimers are going to be reversibly polymerized together. This does take GTP and also occurs in the presence of magnesium. Dimers will aggregate into oligomers, which are sort of the seeds that sort of start as a little baseline for all the other microtubules to start attaching and growing. And this is called nucleation. Then microtubule subunits can start adding at either end, allowing the tube to grow longer. So this is called elongation. How microtubules will assemble in vitro will depend on the concentration of the tubulin dimers. So where you have a concentration where assembly of microtubules and disassembly are balanced, that's called the critical concentration. If the tubulin concentration is higher than the critical concentration, you have more microtubules, you have more tubulin dimers, then the microtubules will grow. And if the concentration is below the critical concentration, you have a lower concentration of tubulin dimers, then the microtubules are going to depolymerize. Microtubules have two different ends, and these two ends are different chemically. And because they're different, one can grow faster or shrink faster than the other end. So the rapidly growing end is called the plus end, and the other end will be called the minus end. You can visualize this by taking basal bodies, and basal bodies are the structures at the base of the cilia with tubulin heterodimers. So the plus and minus ends of microtubules have different critical concentrations. They're adding and subtracting their tubulin at different rates from each other. And if the free tubulin concentration is above the critical concentration for the plus end, so the plus end is going to be adding, but it's below the critical concentration for the minus end is going to be disassembling. Your microtubules will do what's called treadmilling. They'll be adding subunits at the plus end and removing them from the minus end. Because microtubules are important in cell division, they are a target of cancer treatment drugs because one characteristic of cancers is they are dividing more frequently. So paclitaxel and Taxol. Let me give the captioning a time to catch up. Paclitaxel and Taxol. These are drugs which bind tightly to microtubules and stabilize the microtubules. That means that the free tubulin subunits will start to be depleted, and that causes cells that are dividing to arrest during mitosis. So cells will not finish mitosis. It can be used as a cancer treatment. It's especially used for breast cancer. Here are some other chemicals that are used to mess with the cytoskeleton. Colchicine, colcimid, will bind to beta tubulin and inhibit disassembly. Nicotazole will bind to beta tubulin and inhibit polymerization. Vinblastine and vincristin will aggregate tubulin heterodimers and pull them away. And paclitaxel or Taxol, which we discussed, stabilizes microtubules. There are also agents that will affect microfilaments and intermediate filaments that you may want to come back and explore when we're talking about those. Okay, we have a little bit more about microtubules we're going to discuss in a little bit. So come back to our next video and we'll discuss it some more. If you have any more questions, please let me know, and I'll see you later.